Hello everyone, welcome back to our channel. In this week's episode, we are going to be introducing you guys to bioinformatics, such as explaining what exactly it is and how it works. First off, let's start with introductions. My name's Ayushi.
I'm Trisha.
And I'm Connie.
For those who don't know what bioinformatics is, bioinformatics, as related to genetics and genomics, is a scientific subdiscipline that involves using computer technology to collect, store, analyze, and disseminate biological data and information, such as DNA and amino acid sequences or annotations about these sequences.
Scientists and clinicians use databases that organise and index such biological information to increase our understanding of health and disease, and in certain cases, as part of medical care.
One field where bioinformatics is especially useful is in genomics, which can generate vast quantities of information. Genomics is a field of biology focused on studying all the DNA of an organism. So basically, the organism's genome.
In healthcare, clinical bioinformaticians work with a wider team, including clinical geneticists and laboratory scientists, to help provide answers for patients diagnosed with rare disease or cancer.
The main role of clinical bioinformation is to create and use computer programs and software tools to filter large quantities of genomic data, which is usually gathered through next-generation sequencing, a method we will be discussing later on in the podcast, including whole genome sequencing or whole exome sequencing.
I think it's interesting because in doing this, bioinformaticians aim to find meaning in this overwhelming amount of information and provide clinically actionable solutions to help patients. Basically, it is helpful to think of bioinformaticians as librarians. They are responsible for indexing and categorizing data to make it accessible and to find the best and most accurate answer for each specific request. But the question is, how do they do this?
The basic concept of next-generation sequencing, also known as NGS, is that billions of DNA strands are sequenced simultaneously. This method was introduced when the Human Genome Project created a human reference DNA sequence. The basic principle behind NGS is that DNA can be cut into smaller pieces and sequenced. The sequences of these smaller pieces then get assembled based on the reference genome.
A library then gets prepared from the DNA. A library is a collection of short data fragments from a long stretch of data. Libraries get made by cutting the DNA into short pieces of a specified size. Then sequences of DNA called adapters get added to each end of the DNA fragment.
These adapters contain the information needed for sequencing. They also include an index to identify the sample. Finally, any non-bound adapters get removed and the library is complete.
The main sequencing instruments used in NGS are from Illumina, and these instruments use a method called sequencing by synthesis, which also involves sequence clustering, referring to the process of grouping together similar biological sequences.
Once the sequencing of DNA strands are complete, any bad reads get filtered out, such as the bases not being correctly identified. Next, the reads passing the filter get demultiplexed. Demultiplexing uses the attached indexes to identify and sort reads from each sample.
The reads get mapped to the reference genome, and the different reads aligned to the reference genome, overlapping each other. A variant calling algorithm uses the alignment file to look for differences between the genome and the reference, and outputs these to a file known as a variant call file.
To continue on this, the variant file details all the variants that have been discovered. Some will detail many hundreds of thousands of variants, and some will detail only a few, depending on the portion of the genome that has been sequenced. The variants are prioritized using a variety of computational tools and resources, including research and genetic databases, and statistics and modeling.
The prioritized list is then investigated by specialist clinical scientists who examine the variants in the context of other research and literature and information about the patient. Finally, a report is generated that details the findings of the process and is sent to the clinical team to inform the ongoing management of the patient.
Even though we have gone into detail on how bioinformatics is used in geomatics, we also want to talk about proteomics. Proteomics is the study of the structure and function of proteins, and bioinformatics tool can be used to analyze and compare protein sequences through next-generation sequencing. Bioinformatic tools are also used to predict protein structures and functions and identify potential drug targets.
However, despite bioinformatics being extremely useful when it comes to diagnosing patients with diseases, these are still challenges within bioinformatics in the modern day. As research tasks get more and more complex, the required expertise also gets more specialized. This also means the requirement for advanced and higher power of computer resources in the form of specialized software, tools, and also high-performing computer servers.
For example, when trying to predict how a certain protein will function, there are often many different possible outcomes that could occur. In order to try and correctly predict which outcome is most likely to occur, bioinformaticians must have access to large amounts of data and computational power. They must also be able to understand complex biological systems and apply their knowledge in a way that is helpful for industry purposes.
An important challenge that arises when using bioinformatics in the biotechnology industry is the issue of data privacy. Often, companies are hesitant to share data with the outside researchers for fear that it could be used against them in some way. This can make it difficult for bioinformaticians to gain access to the data they need in order to do their work.
Finally, another challenge that can be faced when using bioinformatics in the biotechnology industry is the need for constant updates and upgrades. As new research is published and new technologies are developed, bioinformaticians must be able to keep up with these changes in order to maintain the accuracy of their predictions. This can be a difficult task as it requires a lot of time and effort to stay up to date on all the latest developments.
To summarise this week's podcast, we have introduced you guys to bioinformatics, which is basically just biology and computer science combined, showing how computer science plays a role in nearly all subjects. We have discussed how next-generation sequencing is used particularly in the geometric side of bioinformatics and how the overall process works. We have then also discussed the challenges within bioinformatics.
We hope you've enjoyed listening to this week's podcast and now hopefully have a better understanding of what bioinformatics really is and how it works. To see further content like this, make sure to subscribe to our channel and follow us on Instagram.
We would love to hear any suggestions on future content you would like to see from us, so please comment down below. Thank you for listening.